a publication a few years ago, they looked to see, I think it was in 2003, there was 529 molecules in development for cancer. So they were either in phase 1 or phase 2 or phase 3, 529. And they looked to see a decade later what happened to those 529 molecules. And they found that 45 of them had made it to the market. Well, that's great. 95 were still in development. But 389 molecules were terminated. So as a community, as a biomedical community, we generated 389 molecules and then took them into the clinic and then terminated them. Can you imagine how much money we spent on those 389 molecules? Can you imagine how many people's careers went into those 389 molecules? And can you imagine how many patients were exposed to those molecules and they didn't deliver a new treatment? So it's too risky. In that same study, they looked at if a molecule is in phase 1 for cancer, what's the probability it will make it to the market? The answer was 7.5%. Only less than 1 in 10 molecules in phase 1 for cancer makes it to the market. And they looked to see if a molecule is in phase 3 for cancer, so these are the final registration studies just before you launch the drug. If something's in phase 3, what's the probability it will get to the market? The answer was 33%. So 1 in 3 molecules in phase 3 makes it to the market. So the process is too risky. It's also too slow. And a couple of my colleagues wrote a paper a few years ago where they looked at when did we come up with some data that a particular target would be useful in cancer, and then how long did it take to translate that idea into the clinic? And they estimated that it was anywhere between 6 years and 30 years, 30. The process is too costly, it's too risky, and it's too slow.